I'm Andrew Payne, co-founder and CEO at E11 Bio. We are a focused research organization building tools to unlock scalable mammalian connectomics. I would love to share a little bit of a research update, and thank you, Allison and team, for organizing this awesome workshop. So we are in the era of connectomics now. Really, the fly connectome dropped last year. The fly CNS dropped just a couple of days ago. We're able to produce this data now. It's transforming neuroscience. And to take it to the next level, we need to figure out how to do this in mammals because we are mammals. And if we can do that, it's going to unlock some truly transformative opportunities. It is going to create opportunities to create new therapeutics, including cell therapies that we understand how they integrate into the brain, new targets for BCI. It's going to be able to let us see how prosociality is encoded in the architecture of the brain, and then borrow those ideas for safer and more aligned AI architectures. And if we get the right data, it might even let us simulate specific brains based on the circuitry of those brains. So, any one of these opportunities would transform the world, and connectomics is necessary to unlock all of them. So we really need to figure this out. And the catch is that it's very expensive still. So, if we took the same techniques that got us the fly connectome and we scaled these up to the mouse, it would cost billions of dollars. We need new technology to make this massively cheaper. And I draw inspiration from, the next gen sequencing moment in genomics, where, when the right technology stack came along, the cost of sequencing fell 10,000-fold in 5 years. So these things can happen, but they need to actually happen. So why now? New things are coming along. One of the pieces of the puzzle is likely going to be expansion microscopy. We, seeing Takavoli et al. From Science this year, we can now actually get connectivity out of expansion microscopy samples. This is transformative, but more work is needed. And in particular, whether you are tracing brain circuits with photons or with electrons, tracing is the bottleneck right now and is preventing scale up. And so to illustrate, imagine you're planning a journey across town using a black and white subway map. You're trying to trace these wires. It gets confusing when they cross. The BART is the worst example for this, actually. Everything runs in parallel lines, but imagine London. And as you are tracing these circuits across consecutive cross sections, you make errors. And even with the very best computer vision that we have, those errors are tremendous. It took like 1 million manual edits to the output of the computer vision model to trace 1% of the neurons in the micron's volume. So there is work needed here. And then the second thing is more fundamental. If you drop brain sections as you are slicing and imaging the brain, you cannot trace across the gaps. And between these two things, we have a bottleneck that needs to get resolved. And so, there are different approaches. The thesis that we're testing at E11 Bio, and is the reason for the FRO, is to try to make the neurons do the work. So we are using barcoding borrowed from genomics to give each neuron a different molecular code that lets us trace the neurons more easily and correct these errors. And unlike previous approaches, these are based on molecular codes rather than spectral, but you get the idea. And so to, again, illustrate, if we add color back into this subway map, first of all, it's much easier to trace the subway lines. But second, when we run into one of these gaps where we dropped a brain section, we dropped pulse treat station on the floor, we can use the color to infer across the gap. So this is like self-correcting. It is robust tracing. This is very different than the paradigm where you need continuous images in order to actually successfully trace the neurons. So we had been hard at work on this for a couple of years. We are now very pleased to announce a milestone in our journey. We have released version 1 of a platform for neuron tracing that we call Prism that uses these barcodes. The TLDR from this paper, and I encourage everyone to check it out, the QR code is there, is 1, we now have a barcoding system. It is 750 times more complex than previous spectral barcoding systems. And we think we can scale it further. Every time we come up with a new protein bit, it doubles the barcoding space. We've integrated it with expansion microscopy and AI tracing, so it's not just a barcoding system, it's actually a platform for neuron tracing that people can use now. We show that when you have barcodes, you can trace these neurons more accurately, genetically labeled neurons more accurately. We can get 8x in the first paper. It's improving even as we speak. And critically, you can cross these gaps in space using these barcodes. And we actually have an opportunity to do something entirely new as well. When you have multiple colors and multiple codes, you can bring in molecular information. You can characterize synapses. You can say what synaptic proteins are present. And that is going to be critical if you actually want cell type and synapse type information to simulate these brain circuits. So check it out. A lot of the information is at e11.bio/tech. But the mission of E11 is to accelerate mammalian connectomics. Full stop. And so this is an important step, but it doesn't solve the problem. We have more work to do. And so, the next most important step is to take this platform for neuron tracing and make it fully fledged functional for connectomics. And so that involves combining it with these recent improvements in expansion microscopy that let us actually see the connections between cells and hydrogels. And so that's what we're doing right now. I'm just teasing it with some data here. On the left, you can see the standard expansion microscopy volume, and on the right, you can see prism barcodes layered on top. So this is, the spoilers, this is what's coming next. And we think this is going to be very powerful for scaling up to large volumes, especially when we start bringing in some other exciting opportunities like new microscopes that go much faster than what we can currently use today. We need to be able to combine this with different modalities because, again, if you want to simulate brain circuits, you need more than just the structure. You need, activity recordings. You need to be able to figure out what the transfer functions are. And so that's another milestone on the journey. And critically, if we're going to pay for all of this, we have to start figuring out how connectomics can give back, create these therapeutics, create these BCIs, or at least inform their development. And so that would allow us to create, revenue streams that can be reinvested back into basic R&D. So the Protocol Labs fast R&D approach, that's what we want to see. And so we think there's some really cool opportunities, again, around cell therapies and BCIs. But there's more needed to figure out what all of the opportunities are for connectomics and therapeutics. And so, to wrap it up, connectomics is bottlenecked. We are addressing that bottleneck with self-correcting neuron tracing. We see a clear path to a 100x cost reduction, which is basically a 100x improvement in accuracy, and we've hit 10x already. And we're looking for early adopters and partners and people who want to explore this new connectomics paradigm with us. So please reach out. Now is a good time. Read the preprint. And we're hiring, so if you happen to know somebody who is both into expansion microscopy and is also, a protein detection wizard, we would love to bring them on board. We are also hiring for several other roles now that we're, at this next stage in our journey, so you can check that out. And without further ado, I just want to thank the E11 team. I want to thank our PRISM collaborators on this preprint, including Ed Boyden, Sam Rodriguez, and Jürgen Kornfeld. And I want to thank our funders, Eric and Wendy Schmidt, and James Fickle, the Protocol Labs, and many others who have been supporting us on our journey. Thank you. Let's go to questions.
Ooh. Cool. Just a note that if you guys have job opportunities or anything like it, feel free to drop it also in the WhatsApp chat. That's a good way to for people to easily access it.
Love it.
Max.
Awesome work, as always. I'm curious, what is the plan to transition from mice that you can genetically engineer to humans? Is there anything that you have ready for that?
I think that is going to be, I guess like Andrea said, we have to be realistic. And I think there are a few steps between mice and humans, for example, macaque. And so I would say these technologies develop rapidly. The tech that got us the fly might not be precisely the tech that gets us the mouse. That's our thesis after all. And the tech that gets us the macaque or the human might be different still. And so I would say we should be pragmatic about, incorporating what is the most promising technology at any given time step.
Yeah, more. Thank you.
Well, I guess people always thought there's this canonical local circuit that's universal, right? You repeat the same unit many, many times to cover the whole cortex. But that turns out to be not quite right. So I guess what I'm saying is that even if you cannot, try to do EM level connectome of the whole, say, macaque cortex or human cortex, I think it's very interesting to think about comparing local circuits, let's say V1 versus PFC, in macaque or even in humans. I think you're going to learn a lot that they are very, very different.
Yeah, I think I guess that's more of a comment, but yes, it's comparative connectomics is going to be critical for understanding how these circuits work, because looking at the delta between organisms is in many ways more informative than looking at two different organisms.
especially between areas. So PFC, so why PFC is capable of underlying intelligence, cognition, rather than sensory processing, right?
Yeah.
There's something, that we need to learn about. So, this kind of tool, I think, would be really useful for that. Thank you.
Thanks a lot. Thank you so much. We have to leave it at that. But we have the long breakouts.